In this class, I will discuss about B cell activations, how B cell get activated. So at first, you must know that B cell can be activated by two ways. The first is T cell dependent B cell activation, and the second way is T cell independent B cell activation. Some B cells can be activated by T cells. These B cells are called B2 B cells. This cells is also known as follicular B cell. Why it is called follicular B cells? Because this type of B cells, B2 B cells, are present in the follicle of spleen and lymph node. Both these secondary lymphoid organ have follicle, and those B cells that reside in the follicle of spleen and lymph node are called B2 B cells or follicular B cells. These B cells actually activated by T cells, and there are some subset of B cells that do not require T cell help. The cells are B1 B cells. These cells are present in the peritoneal cavity, and another cell that is called marginal zone B cell. These B cells are present in the marginal zone of spleen, and there is difference between their antigen recognitions. Those B cells that are activated by T cells, these B cells actually recognize only protein antigen, and here they actually recognize polysaccharide or repeated molecules. Those molecules have repeat unit, and polysaccharide like LPS, lipopolysaccharide of negative gram bacteria. So they can recognize polysaccharide, not antigen. And there is many thing occur if the B cell is activated by the T cell or not. That is, first is when the B cell gets activated by the help of T cells, they become long-lived plasma cells. They can become long-lived plasma cells. That is the first thing occur. Second thing, they can produce memory cell. Third thing, they can go through class switching. That means they can produce different type of antibody, IgG, IgA, IgE, and their subtypes. There are IgG1, IgG2, IgG3, IgG4. These are subtypes. So class switching. And the fourth thing is they go through a process called somatic hypermutations, by which actually thing occurs that is called affinity maturations, that actually occurs in antibody. So affinity maturation of antibody. What is affinity maturation? When these B2 B cells activated by the help of T cells, they produce antibody that can recognize the antigen with high affinity. So affinity maturation means the antibody can bind with the antigen with high affinity. But here you can see, if there is no T cell dependency or help, then what happens? The cell becomes short-lived plasma cells. They do not produce memory B cell. So cross 3 is class switching do not occur, so they cannot produce IgG, IgA, IgE. They can only produce IgM type of antibody. So there is a limitation. And another thing is they do not go through somatic hypermutations, and so that they do not produce antibody with high affinity. How much T cells are important for the B cell activations? So in this class I will talk about B2 B cells, about the activation of B2 B cell. Okay, as I have already mentioned that for the proper activation of B cells, T cell is important, but the T cell that actually activate the B cell, the T cell must be activated at first. So what happens if I give you a overall scenario? What happens? Let's assume there is a tissue site. In the tissue site, there is already some sentinels are present called dendritic cells. These dendritic cells express a type of receptor called pattern recognition receptors. This pattern recognition receptor can identify pathogen signature molecule. That means some molecules are present on pathogen, they can recognize. So pattern recognition receptors actually identify PAM, pathogen associated molecular pattern. In case of many pathogens, they have double strand RNA, they have specific protein like flagella that is not present in eukaryotic cells. Flagellin is only present in the flagella. So likewise, like LPS, lipopolysaccharide, not present in eukaryotic cells, present only gram-positive cells, gram-negative cells. Gram-positive cells have lipoteichoic acids. So these are the pathogen-associated molecular pattern, means some signature molecule of pathogen. This pattern or signature molecule of the pathogen can be recognized by pattern recognition receptors. These are the pattern recognition receptors expressed by the dendritic cells. Example of these pattern recognition receptors are toll-like receptor, knot-like receptor, retinoic acid-like receptor, C-type lectin receptor, and there are many different type of toll-like receptor, knot-like receptors. So let's assume now this in the tissue side there is infections. These are the pathogen. So this dendritic cell can sense the pathogen, presence of the pathogen, and the dendritic cell can engulf the pathogen by a process called phagocytosis. When this dendritic cells phagocytose any pathogen, it actually process the pathogen and actually present the fragment of the pathogen by MHC2 molecule. That's why this dendritic cell is called professional antigen presenting cell. Some dendritic cells leave the infected tissue sites. So this is infected tissue site. Some dendritic cells leave this infected tissue sites to the nearest lymph node. But if there is any pathogen present in the blood directly, what happens? The pathogen is actually reach to a secondary lymphoid organ called spleen directly. The pathogen are trapped. This pathogen go through the blood and reach into the spleen. So they are trapped in the spleen, and in the spleen there is already some dendritic cells are present that can recognize this pathogen by the help of pattern recognition receptors. In the lymph node and in the spleen, naive T cells actually patrol. Naive means those T cells have not yet recognized any pathogen or antigen. That means the inactive cells. So inactive cells, inactive T cells actually patrol through this lymph node. So there are T cells. Here I am only mentioning T helper cells, not the cytotoxic T cells, because for the activation of B cells, helper cell is important. So I am talking about helper cell. Let's assume this is a T helper cell that is naive or inactive in conditions. Naive T cells. So many naive T cells patrol through the spleen and the lymph node. If they found that a dendritic cell is present that is presenting pathogen antigen, these dendritic cells activate the T helper, naive T helper cell into effector T helper cells. Now these T helper cells become effector. That means now these cells become activated. So I can write activated. I am not going to details how dendritic cell activate naive T cells, because I have made a separate video on which I have discussed about how dendritic cells activate naive cells. Here I want to mention you that the place where this dendritic cell and the TH naive cells actually interact, in case of lymph node, the area is called paracortex. This paracortex is actually the T cell zone. So I can write that this paracortex is T cell zone where T cell actually reside. But in case of spleen, this region is called PALS, periarteriolar lymphoid sheath. I am writing here. PALS means periarteriolar lymphoid sheath. In case of spleen, this is the region where T cell actually present. So I can write that this is the T cell zone, and another important cell actually activated, that is called, for the activation of B cells, that is very important, another cell activated, that is called T follicular helper cell. This cell is also produced and activated by the dendritic cell. This is very important for the B cell activation. If the T cell receptor and MHC interaction is very high strength, if any T cells recognize the MHC molecule with high strength, that cell become the T follicular helper cells. Now what happens? In the spleen and in the lymph node, there is a region I have already mentioned, follicle. There is a follicle region. Naive B cells are present. Many naive B cells are present. I am showing you one. Naive means those B cells are inactive in conditions. In case of lymph node, the follicle and the paracortex are very close to each other, and also in case of spleen, the PALS and the follicle is very close to each other. This is very significant because in both these secondary lymphoid organ, the T cell zone and this is the B cell zone are very close to each other, because that immune system know that for the activation of B cell, T cell is very important. So they are not present in distant, they are very close to each other. Okay, so this is the B cell zone. I can write that this is B cell zone. Now what happens when these effector cells get activated or naive cells get activated? Some B cells actually come out. These naive B cells come out and actually interact with the activated T cells to get activated. Okay, let's assume this is the effector T cells, T helper cell. Okay, and this is the naive B cell. When B cell is naive in conditions or inactive in conditions, they express two antibody, that is membrane bound in conditions. One is called IgM and another is IgD. These two antibody actually called togetherly called B cell receptor. As the T cell have T cell receptor, this B cell have B cell receptor. That means IgM and IgD antibody, that is membrane bound in conditions. Naive B cells can recognize pathogen or antigen with the help of this receptor or antibody. And for the B cell activation, this is the first signal. These interactions, antibody and the antigen interaction, is the first signal for their activations. When this antibody or B cell receptor attach with antigen or pathogen, it actually perform a process called receptor mediated endocytosis. So these cells actually perform a mechanism called receptor mediated endocytosis. By this process, this B cell actually engulf the antigen along with the antibody and process the antigen and present the antigen fragment with the help of MHC2 molecules. So let me draw a MHC molecule. As we all know that B cell is a professional antigen presenting cells, so it has a class 2 MHC molecule. Now what happens, this T cell have T cell receptor that can recognize, this is T cell receptor that can recognize MHC2 and peptide complex. And there is another thing in the T cell, that is a molecule called CD28. This is a receptor. In case of T cell, there is a receptor called CD28. This is a co-stimulatory molecule receptor. This molecule bind with the ligand that is known as CD80, CD86. The activated T cells activate the naive B cells. Okay, so this is the first signal. Then CD28 attach with the CD80 and 86. Another thing is this activated effector T cells have a ligand that is known as CD40L. This is very important. I am mentioning you. This B cell have a receptor like structure that can recognize the CD40L, that is called CD40. When the CD40L and the CD40 interact, this B cell get the second signal for its activations. And the last thing for the activation of B cells, these effector T cells, it could be Th1, Th2, these cells actually secrete cytokines. So this is the last thing that actually activate the B cells. So they secrete cytokine, and these B cells have receptor for that cytokine, and this is the third signals that actually activate these B cells. When these three signals actually integrate into these cells become activated and many things occur. When this B cells get activated, it actually at first become short-lived plasma cells. It starts to produce some antibody, different type of antibody, so class switching occur, but the class switching is not extensive. And the third thing is somatic hypermutations do not occur still now. So this is the first time the B cell get activated by the activated T cells. But for the production of long-lived plasma cells, as I have already mentioned, this is the B2 B cells. So B2 B cells can produce long-lived plasma cells, memory B cells, and the class switching occur very extensively in case of B2 B cells, and they go through somatic hypermutations. But for the first time activations, for the first time B cell activations, these things actually do not occur. What happens? These short-lived plasma cells actually leave the spleen and the lymph node to the infected tissue site. So the cells actually go and actually reach to the infected tissue site. for the elimination of the pathogen. But what happens? Some these short-lived plasma cells re-enter into the follicle. Okay, re-enter into the follicle, and another thing occur, that is, so T follicular helper cells enter and these activated B cells enter. Now what happens? This is called follicle. This is also known as primary follicle. So at first it was primary follicle. When these short-lived plasma cells re-enter and the T follicular helper cell re-enter, now these primary follicles become secondary follicles, and the secondary follicle is actually very importantly known as germinal center. In the germinal center, this activated B cells gets activated another time by the help of T follicular helper cells. That is, for it is called follicular helper cells, because it actually enter into the follicle and help the B cells to become. Now this, when this B cell and T follicular helper cell interact, they interact each other, and one important interactions, there are all these interaction occurs, but the main interaction that occur between this T follicular helper cell and B cell is the CD40 ligand and CD40 interaction. This is the main interactions that occur and actually result in the ultimate phenomenon of these B cells. That is, when this B cell and T helper cells interact, they become, this B cell become long-lived plasma cells. They can become memory cell. Some plasma cells become memory cells. Then class switching starts to occur very extensively, and the fourth thing is somatic hypermutation occur. Somatic hypermutation occur. So affinity maturation is the ultimate result. That means now these B cells are producing antibody that can recognize the same antigen with high affinity. At first it was not so high affinity, but now it can produce high affinity antibody. So all these things actually occur due to this molecule CD40 ligand and CD40 interactions. Why it is so important for the B cell activations? Because for this CD40L and CD40 interaction, one enzyme is activated. One enzyme actually gets activated, that is called AID. The full form of AID is activation induced cytidine deaminase, and this enzyme is responsible for class switching to occur extensively and somatic hypermutations. And there is another cells called follicular dendritic cells. This cells is also present in the follicle. This cell also help in the formation of germinal center.